Welcome, everyone. I remember coming here back in, I think, 2018 to one of my first conferences here. And I remember sitting in one of those seats and listening to one of these talks and thinking, oh boy, I really hope I get to do one of these cool presentations today. So that's what I will be doing today. I would like to dedicate this presentation to my former Szent teacher, Mr. József Gőzt, who I believe is here somewhere. And I think that's about it. Let's begin. Sugars against the flu, an introduction to targeted protein degradation. What in the seven hells could that possibly mean? Before we start talking about the topic of my research in earnest, I think we need to clear up some things. First, we are going to talk about sugars, then we are going to talk about the flu, and then we will talk about targeted protein degradation, and then we will bring these three concepts together and actually talk about what I do in the lab. For those of you who are not really into chemistry, I'm sorry, I tried to include as little chemistry in this presentation as I could, but that's a bit difficult to do when you work in a chemical lab. So, sugars. What are sugars? In everyday life, we refer to sugar, we mean the crystalline sweet stuff that half the world is addicted to. But chemically, sugars are small molecule carbohydrates, meaning that they have the general formula CH2ON, where N denotes an integer. This is a very loose definition, and deviations from this general formula exist, as you will see in a second. But I brought you three examples to show you how diverse sugars are. I'm sure that most of you are familiar at this point with glucose, which is the most abundant monosaccharide in the world. Monosaccharide meaning it has one sugar unit. And you may also be familiar with galactose, which is its cousin. They both have the same formula C6H12O6, meaning they are both composed of 6 carbon, 12 hydrogen, and 6 oxygen atoms. But one of the hydroxyl groups is flipped in galactose, denoted in blue, meaning those of you who have not had organic chemistry yet, the red line means that it's pointing away from you, and the blue line means that it's pointing towards you. It has a different spatial orientation. And because of this very small difference in structure, they have slightly different biological roles. I think this is best demonstrated if we take 2 glucose and 1 glucose and 1 galactose molecule and combine them together into disaccharides. Disaccharides meaning they have 2 sugar units. Now you get maltose and lactose, which are slightly different again in structure. If you take one look, you can't really tell the difference, but there are very small configurational differences, stereochemical differences between the two. And due to this, they have different biological effects. If you give someone who has lactose intolerance lactose without their lactase, they will have general gastrointestinal distress and stomach aches and diarrhea. But if you give them maltose, which is very similar and is broken down by a different enzyme, maltase, they are going to be completely fine. Small stereochemical difference, but leads to a very different biological response. Then we have cytidine, which is a nucleoside, which means it's a sugar connected to a nucleobase, and it's one of the letters of the genetic code in RNA. RNA is the cousin of DNA that I'm sure most of you are already familiar with. Instead of a double helix, RNA forms a single helix, where the nucleobases denote the genetic information in the form of these letters, and the sugar phosphate chain creates the backbone. Now, what's interesting, if you take cytidine and you flip one hydroxyl group, as in the case of glucose and galactose, you get cytarabine, which is a drug molecule which is used in the treatment of leukemia. Very small difference, but it leads to a vastly different biological response. As a final example, this tetrasaccharide is the type A antigen, which is responsible for type A blood. It is found on the surface of red blood cells, and it is very, very similar to the B antigen. For those of you who can't see the difference, it's these small groups. And if you compare it to the size of a red blood cell, it's almost unimaginably small. It's like trying to see the single grain of sand on the surface of the skin of an elephant. And our immune cells can tell the difference between these small differences in the saccharides on our cells and use that as identification tags. So if you give someone type B blood, but they have type A blood, that could lead to disastrous consequences because their immune cells will recognize these differences of a few atoms as different, and they will attack those cells. So this is good to know. So once again, to sum it up, small, subtle chemical differences lead to very different biological responses. This is not a unique trait to carbohydrates. This is found in all biomolecules, but it is very apparent in carbohydrates. So besides sources of energy, sugars also serve as components of nucleic acid and identification tags in cell recognition and communication. And they also serve as drugs. Sugars and the derivatives are important drugs. I have shown you cytarabine. There is also topiramate, which is used in the treatment of epilepsy. Fondaparinux, which is a pentasaccharide, meaning it has 5 sugar units, used as a blood thinner, a blood coagulation inhibitor drug. Then we have digoxin, used in the treatment of heart failure, dapagliflozin, which is used in the treatment of type 2 diabetes. And then we have zanamivir, which is used in the treatment of influenza, which brings us to part 2. Let's talk about influenza a little bit before we talk about zanamivir. I'm sure most of you here have already had influenza at some point in your lives. It's one of the most common viral infections worldwide, affecting hundreds of millions of people each year. It's caused, obviously, by influenza viruses. And when someone gets influenza, we don't usually give them antivirals, meaning medication that specifically targets the virus. We give them symptomatic treatment, lower their fever, and give them hydration. But antivirals are used usually in severe cases. If you take a look at the life cycle of influenza viruses, obviously when you have someone who is infected with influenza, when they cough, they can release respiratory droplets which contain influenza particles. These influenza viral particles enter your respiratory system and cause the infection. They infect cells by latching onto glycoproteins on the surface of cells, which are proteins which have polysaccharide chains attached, denoted by those small hexagons. And many of these glycoproteins, which also serve as identification tags, as shown in the case of the type A antigen on the surface of red blood cells, many of these contain sialic acid, which is a special type of sugar that you can see here. One of the proteins of influenza, hemagglutinin, denoted by the red protein, can attach onto the sialic acid, grab onto it, and use that to enter the cell. Once it enters the cell, it releases its genetic information in the form of RNA molecules. These RNA molecules are replicated, and they are also transcribed to protein. The virus multiplies and uses the cell's framework as a way to make its own descendants, its own viral proteins. Then these components assemble and give a new viral particle. Then this viral particle tries to leave the cell. But sometimes it cannot. Imagine that you are walking out of a room and your belt ring or your shirt gets stuck on the door handle. I'm sure this has happened to many of you before. Something similar can happen to influenza because the hemagglutinin, which allowed the entry into the cell, now also gets stuck on the sialic acid, and it cannot go and infect other cells. How can the influenza virus avoid this? It's smart. It's not smart because it's just a virus. It's just a bunch of molecules. But what it does is very smart. Let's take another look at hemagglutinin. It also has a pair called neuraminidase. Neuraminidase also can bind sialic acid, but instead of simply attaching onto it, it acts as a pair of scissors and cuts off the sialic acid at the end of the glycoprotein. The sialic acid is cleaved off, and now we have a modified glycoprotein. This modified glycoprotein does not have a strong interaction with the hemagglutinin, and because of this, lack of interaction, it will not create a strong binding, and the virus can go and infect other cells. This is good for the virus, but it's obviously bad for us, because it means the infection will go on to other cells. What we can do is use a molecule which will get stuck between the blades of this molecular scissor, which will not allow the sialic acid to fit into the neuraminidase. And because of this, it will get stuck on the hemagglutinin, and this will slow the spread of the virus. That's great for us. This molecule, as I mentioned before, is zanamivir. It also has a cousin called oseltamivir. They are neuraminidase inhibitors. Due to being close structural analogs, you can see that they are similar in structure to sialic acid. They can inhibit the spread of the viruses and slow the spread of viruses. And because all they do is slow this spread, they are most effective when used in the early stages of influenza, but there are not so many viral particles in our system. Which begs the question, can we make them more effective? What if there was a way to modify these molecules so the neuraminidase protein isn't simply inhibited, blocked, but fully destroyed inside the cell? And this would stop the spread of the virus completely, because a virus is like a jigsaw puzzle, and if there is one piece missing, if we destroy one piece, the neuraminidase protein, then the virus will be ineffective. It cannot form a complete virus. So, yes, there is a way to do that, and it's called targeted protein degradation. Targeted protein degradation is a new approach in drug design. Instead of simply inhibiting molecules, we induce their complete destruction and degradation. Since there is no protein, it cannot fulfill the action. There are, at this point, like 2 dozen different ways to do it, but I will only be talking about one approach today, which is called the PROTAC modality, which is short for proteolysis targeting chimeras. Now, imagine that you have a protein which is damaged and misfolded, and because of that, it's dangerous/useless, and we want to get rid of it. An E3 ligase, which is a ubiquitinating enzyme, will come along with a small protein called a ubiquitin. And imagine that you have a jerk friend who comes along and pats you on the back and puts a sticky note on your back that says, "Kick me." This is what the E3 ligase does. It puts a tag onto this damaged protein that basically tells the cell that this protein needs to be destroyed. Now, this damaged misfolded protein, which is now ubiquitinated, undergoes the same fate that Pedro Pascal's character in the Kingsman 2 Golden Circle, Agent Whiskey, undergoes at the end of the movie. It is shredded to pieces by a meat grinder called the proteasome. So it's destroyed now. That's great. How can we take advantage of this intrinsic system built into our cells? What we can do is we take our target protein, our E3 ligase, and we design ligands for it. Ligands are like keys that fit into these small binding pockets on the surface of proteins. Okay, so we have our ligands. This is not interesting. What's interesting is what happens when you link these molecules together by any type of linker, basically. Then you get a PROTAC molecule. What happens now is that these proteins are brought in close proximity. This proximity basically is enough for the ubiquitination. Because of this closeness, the E3 ligase will ubiquitinate our target protein, which will induce its degradation. And our protein is now just another Agent Whiskey in the history of proteins. Now, our goal, specifically, is to make zanamivir-based PROTACs, meaning molecules which bind to viral neuraminidase, but instead of simply inhibiting it, it will also induce its intracellular degradation. So, when I had this idea, and it was approved by my mentor, I was told to make a plan. First thing you do when you get an idea in research and you want to pursue it is if you read into the literature and find out if anyone has done something similar. Hopefully not the exact same thing, because then it's a useless idea at that point. But if someone has done something similar, then you can use that as inspiration, and you can use their findings to further your own. So we did find an article which described the synthesis of oseltamivir-based protex, which is, if you remember, the cousin of zanamivir. So now we know it's technically a feasible approach. Then we just keep reading, write down all our ideas, and figure out the most obvious problems. One of the most obvious problems that we had is figuring out where we would attach the linker, which would be the connection point on this zanamivir molecule, which is the blue hexagon in this situation. Because the zanamivir acts as a piece of puzzle or a key fitting into the neuraminidase. If you modify the wrong functional group, it will not be able to fit into this puzzle anymore. And if there is no binding, then it becomes an ineffective molecule. It's like trying to fit a piece of a jigsaw puzzle, and if you modify the upper side, left side, or bottom side, it won't be able to fit anymore. We want to find a side, which is the right side, which can be modified without affecting binding. So we read up the literature, checked out which modifications result in decreased binding, and we found that, for example, the carboxylate modification is very easy synthetically. Like it would have been so easy if you could do that, but unfortunately, the modification here results in loss of activity in most cases. The guanidine group is a bit more difficult synthetically, but the activity is somewhat retained. That's an okay approach. What we found was that the 7-OH is the ideal position for modification because the literature data indicates that there is minimal interference with binding. Unfortunately for us, that is also the most difficult approach synthetically, obviously. So then we draw up some molecules. Now we know what we want to do and where we want to modify the zanamivir. If you have seen the series Breaking Bad, which has an aspiring chemist and lovely young man called Jesse Pinkman, he refers to making amphetamines, so chemical synthesis. He says something along the lines of, this ain't chemistry, this is cooking. Cooking is art. And chemistry, he is very close to the truth because chemistry is very much like chemistry. Chemistry is very much like cooking. If we compare this molecule to a pizza, then we have our pizza base, our sauces, our toppings. But to make a pizza, you have to follow a specific recipe, and you have to start from flour and water. And you have to knead the dough, and you have to cook the pizza at the right temperature, add just the right amount of salt. You need very specific conditions to get the perfect pizza. And chemical synthesis is like that. I will not be talking about the steps today. This is the synthetic plant that we are currently working on to make one of these molecules. So we started this synthesis, and what's the next step? Now, you watch as the plant does not work for some reason, any reason. Then, instead of giving up, which is an option that I have considered a couple of times in the past years, but you make tweaks, you figure out what went wrong, you try again, and then you watch as it doesn't work again, but then you just keep repeating steps 4 to 5 until finally something works, which is just how, not just chemistry, it's how research works. So that's what we are working on right now. Conclusion. Influenza is a viral infection that can be treated with special sugars, cool sugars called neuraminidase inhibitors, like zanamivir, oseltamivir, there's also leronamivir, there are a couple. And if you turn these molecules into targeted protein degraders, protex, then we can potentially have molecules which have better antiviral action. The synthesis is currently underway. Our take-home message is sugars are cool, chemistry is cool, and research work is grueling, difficult, annoying, but it is so worth it to have a molecule that no one else has in the whole wide world in your hand, finally, after weeks of work. That's just gratifying. And if it has biological activity, even better. So, thank you for your attention, and I hope some of you at least will pursue a chemical career. Thank you.
Thank you very much for this nice, interesting, and relaxed presentation. Please meet us in the middle for an amazing photo.